Yeah. You're okay. You ready? It's working now.
Ok,
Tá pronto, tá pronto.
okay. So I'm going to present Catherine McFadden, or Cassie, as she's known. Cassie is here from the Capri Sprint program. She's going to be here for 3 weeks. It's her first week, and she is giving a course this week. And then we're going to be going into the field and doing 2 weeks of field work in Rio and in Bahia as well. So, so Cassie, she got her PhD in zoology from the University of Washington, and her PhD was on growth, feeding biology, and clonal demography of fragmenting soft coral. And that's kind of was what she's been, I think, doing ever since, right? I'm sure she's going to talk much more about it, but she's worked all around the world with the taxonomy and phylogenetics of octocorals and soft corals, and she has hundreds of publications. I think literally hundreds of publications on the subject.
Not plural.
Well, yeah. And she was I met her first when she was a NATO postdoc at the where I did my PhD. She was a postdoc, which was a marine biological station, which was part of the University of Liverpool on the Isle of Man, which is between England, Scotland, and Ireland in the middle of the sea. And so years later, we got in touch through a mutual interest in octocorals, and especially in my case with invasive species, which she's also been working on. And she's now the Vivian and D. Kenneth Baker Professor of Biology at Harvey Mudd College in California. So it's a real pleasure to have Kathy here, and I'm looking forward to a great presentation. Thank you very much for being here, Kathy.
Thanks. Yeah, perfect. Okay, thank you. Well, thanks, Joel, for the introduction, and thank you all for inviting me to speak about my work while I'm here. I started working on a soft coral as a PhD student just because I was interested in the unusual things it was doing about its reproduction, and somehow I ended up getting stuck my whole career studying soft corals. I kept trying to get away from soft corals and into other systems, and kept coming back to soft corals. So when I sent my title to Joel last week, it was Coral Species, Speciation, and Trait Evolution, and the things we're learning about those from phylogenomic approaches. I was a little ambitious, I think, and as I put together the talk and realized unless we wanted to be here all afternoon, I needed to cut down some of this. So I won't talk very much about the speciation part of it, but we'll focus on more on the phylogenetics and trait evolution and what we're learning about from recently developed molecular techniques. So the corals belong to a subphylum of the Cnidaria. It's called Anthozoa. And there are two major groups in the Anthozoa, the class Hexacoralia, and this includes all of the sea anemones and black corals, the deep sea, and also the very familiar scleractinian or stony corals that form shallow water coral reefs. Those are all hexacoral. And the other class of Anthozoa are the octocorals, and this is the group that I specialize in and will be talking about today. So the octocorals, there are about 3,500 species, we think. One of the problems is we don't know how many there really are. There are many that are undescribed. There are many that have been described with multiple names, and trying to figure out who the species are, how many are there, where are they, is one of the main things that I do. This is just a selection of shallow water octocorals from throughout the world. They're very common and ecologically important on shallow water coral reefs, but also in the deep sea and in other parts of the world. So just to illustrate some of this ecological importance, this is a shallow coral reef flat in the Red Sea. And almost everything you see in this photo is soft coral. It's not the hard corals that are building the reef. There's a few of them here and there, but this is all soft coral that's dominating this reef, so they can be very important space occupiers on reefs. This is what much of the Caribbean looks like right now. Okay, so as the stony corals have been decreasing in abundance as a result of environmental impacts like climate change, reef communities are shifting to be dominated by these Gorgonian sea fans, which are also octocorals, and we now have these forests of corals where there had been reefs of stony corals. And these forests also are common and important in the deep sea. So this is a deep sea community, several thousand meters deep. And what we have is anywhere there's a small amount of hard substrate is colonized with these corals. This is a mix of stony corals and octocorals, and they're providing three-dimensional structure to this community, again, like a forest. Without the corals, here is just a two-dimensional mudflat, and there are many other species associated with these coral communities. You can see lots of brittle stars and other invertebrates and often fish and things that are living on these corals. So the octocorals, as well as the stony corals, are very important foundation species for many of these ecological communities. They're building the substrate that other organisms rely on and providing that three-dimensional structure to the community. But the octocorals are also impacted by the same problems that are impacting stony corals around the world. So coral bleaching is a big problem that has contributed to mortality worldwide of stony corals. The soft corals also experience bleaching. So like the stony corals, they have symbiotic algae in their tissues that photosynthesize, rely on, for much of their nutrition, they rely on the products of that photosynthesis. And when that symbiosis breaks down in response to warm water or other environmental stressors, they lose their symbionts and very quickly die. So this is a real problem that's affecting both octocorals and stony corals around the world. So we've been trying to, the stony corals are much better studied, much better understood than these octocorals. And what we've been trying to do is basically increase our knowledge of these octocorals, because they are so important and may be even more important in the future as the stony coral reefs are declining. So what I want to do today is start by telling you a little bit more about the octocorals and how we classified them up until very recently. Lessons that we learned from the earlier studies using molecular methods that suggested to us that our classification and our understanding of evolutionary relationships and evolution of traits was flawed. I'll talk a little about some of the methods that we've developed and been using over the last 5 to 10 years to try to come to better understanding of the evolutionary relationships and species boundaries in this group, and then talk a little bit about how we revise the classification and our new understanding of how some of these traits have evolved. And finally, I'll say a little bit at the end about the work we're doing on species boundaries and trying to understand how many species are there, who are they, and where they are. So for most of the 20th century, the octocorals were divided into 3 orders, and this was based on differences in their morphology, quite quite large differences in their morphology. So we had an order called Panattulaceae, which are sea pens, and about 200 species in 14 families. Another order, Helioporacea, called blue coral, very little diversity, only a few species in 2 families in this group, and everything else, over 3,000 species in this order Alcyanacea. So to show you the differences between these and why they were put in different orders, this is the blue coral, and this is the only octocoral that produces a skeleton of calcium carbonate, and in particular aragonite, the same form of calcium carbonate that the stony corals produce. So this is a true reef-building coral. There's some very large reefs in the Western Pacific that are made almost entirely by this octocoral. So it forms true reef structures. And it's the only octocoral that does that. The sea pens are also very unique morphologically among octocorals. They are colonies that are highly specialized to live in soft sediment. And the colony takes on this very, very specialized form and has different parts of the colony are differentiated for different functions. So sea pens have this lower part called a peduncle, which is basically an inflatable bag that they can use to anchor themselves in the soft sediment. And the rest of the colony sticks up into the water, called the rachis, and the polyps that it uses for feeding are arranged around the rachis, and they're often on these structures that we call leaves. So it's very highly specialized morphology that all of these sea pens have. So different order. And then everything else was in this one order Alcyanacea, and this includes 2 different major morphologies. What I would call the true soft corals, and these are colonies that have no internal skeletal axis. So this can be quite tall, branched animal, but there's nothing inside it except water. So they can contract down to a very small size, and when the tissue dies, there's nothing left. Right? The other group of Alcyanacea are what we refer to as gorgonians or sea fans, and these do have an internal skeletal axis. So these are typically upright, branched, tree-like colonies, and inside the branches are solid rods of either calcium carbonate or protein to support them. So 2 distinctions: soft coral, gorgonian. 3,000 species, all in one order. So over time, there were attempts to try to divide this order into suborders that made sense based on differences in morphology. So again, for most of the 20th century, we had 5 or 6 suborders within Alcyanacea, 3 suborders for the gorgonians, so things with an axis, and those suborders were differed by what the axis is made of. So this group, Chalcozonia, the axis is solid calcium carbonate, it's a rod of calcium carbonate. In the Holaxonia, the axis is made of a protein called gorgonin, which is how they get the name gorgonian, is they have gorgonin is a protein that's very similar to horn. And the third group of gorgonians and Scleraxonia have an axis that was often a mixture of protein and microscopic crystals of calcium carbonate that are called sclerites. And these sclerites are typically present in the soft tissue of all of these octocorals. In addition to a skeletal axis, they have these little crystals, but in this case, the axis is made of those crystals that are fused together or held together with protein or other calcium carbonate. So those were 3 different groups based on what's the skeletal axis made out of. The soft corals, which don't have an axis, are divided into 2 suborders. Alcyaneana, where the polyps are in a big mass of tissue called coenenchyme, and the stolonifera, where the polyps are just connected by very thin membrane of tissue. So if we look at those as stoloniferan, the colony looks like this. So there's polyps, which have a mouth and tentacles they use for feeding, and then the body of the polyp. And the polyps are just connected together by either a thin membrane of tissue or a tube of tissue called a stolon. And this is different from the other Alcyoniana soft corals, where again we have the same polyps, but they are in this big mass of tissue that can be very thick, and the polyps can retract down into that mass of tissue. Okay? And then these differ from the gorgonians, and a gorgonian also has polyps that are in tissue, usually a thin layer of tissue, but that tissue is wrapped around the axis. So here's the axis is here, the solid rod of calcium carbonate or gorgonian, tissue around it, and polyps in that tissue. So those are the major morphological differences between these groups of Alcyonasians. Okay, so that's how we classified them, again, based on these pretty major differences in their structure. And when we first started doing molecular phylogenetic work around 2000 or so, we discovered that this classification system did not reflect the evolutionary relationships of these different groups. This is the first molecular phylogeny that was done for octocorals, and it's probably hard to see there, but these are the different skeletal types of different suborders I've just shown you with little symbols indicating each one. And if you see from the tree, those little symbols are spread all throughout the tree. So it's clear that these different suborders, not all of the species that belong to that same suborder have the same common ancestor. This is a tree that my lab published a few years later using several mitochondrial genes. And in this case, the different skeleton types are shown with colors. Again, you can see the same thing. So red here are gorgonians with a proteinaceous axis. You see they're all throughout the tree. Like blue are the Alcyonian soft corals. Yellow are some of these stoloniferous groups. They're all mixed up together, right? So the different suborders and orders are not coming out as monophyletic groups or clades. What we did see, though, which was unexpected, is that the octocorals divided into two major groups. So we have one major group here, and then a second group, the clade here. And the different types of skeletons are found in both of those groups. The only major difference between the groups is that most, but not all, of the species in this group are shallow water, and most of the species in this group are deep water. They're exceptions. So we had tried to go further with this phylogeny. It was clear that we had these two major clades. We don't see the three orders, Penatulacea, Helioporacea, and Alcyonacea, separating as three distinct groups. And at the level of families, most of the families were also not monophyletic. So there were species belonging to the same family that were spread all through the tree. The problem we had in trying to better understand these evolutionary relationships is that while we see these two main groups, within each group, there's very little resolution along what we call the backbone of the tree to figure out how groups that are in there are actually related to one another. So this was a tree. We had a project through the 2000s to try to, well, the goal was to understand the phylogeny of all of the cnidarians, over 10,000 species. We're going to sequence them and have the whole evolutionary tree. And we sequenced what for the time was a very large amount of data, 18S, 28S, and 16S ribosomal DNA, And this is the tree we got. We never published it because it didn't give us any more information than what we already had. So again, we see one major group, and it includes soft corals, stoloniferans, the gorgonians with proteinaceous axis and other things. Another major group here with some of the other groups in it, but we just don't have enough resolution to tell how these different groups within here are really related to one another. So the other, at a lower level of the phylogeny, again, we have these problems with families where our understanding of families, and again, things were grouped into families based on morphological characteristics, similarities they shared in a colony growth form, skeleton, and when we look at the phylogenetic distribution of families, we see the same problem. These are two examples I worked on. This is a family called Clavulariidae, which included most of the species with that stoloniferous growth morphology, just connected by a membrane of tissue. And in this tree, everything in a gray box, here, belong to that family Clavulariidae. Okay, so again, the Clavulariidae don't have one single common ancestor. That growth form appears to have evolved many times throughout the group. So things with that growth morphology are not closely related to one another. This is another family Alcyonidae, which was a family of many of those big fleshy soft corals. And everything in this tree that's red or blue was in Alcyonidae. Again, we see members of Alcyonidae all throughout the tree. So, and this is true, these are probably the two most extreme families as far as that goes, but almost every family we looked at was not monophyletic, so there was not a single common ancestor of the species and genera that have been put in that family. Again, suggesting that many of these morphological traits have probably evolved multiple times in the group. So we were stuck here for a long time because we were trying different genes to get better resolution and understand the phylogeny better. And every gene we tried, we still had the same problem of not enough resolution along the backbone of the tree to really understand how things were related to one another. So I'm going to shift here for a minute and talk about some of the different methods that have been developed and used over the last probably 5 to 10 years for getting lots more genetic data. And these are all what we call phylogenomic methods. So they're methods by which we can sequence the entire genome and use data from many, many, many more genes than if we're just looking at one gene at a time or a few genes. So high throughput sequencing or next generation sequencing is the technology that allows us to sequence whole genomes very, very quickly and easily. And while we can do that, for addressing these types of questions, we don't need the whole genome. Okay, that's more data than we need. So a number of techniques that have been developed that are called reduced representation methods, where we sequence some subset of genes in the genome. You don't need the whole genome, you just need a couple hundred or thousand genes, be nice. So just to list some of the different methods of high throughput sequencing that are being used and applied to questions in phylogeny and often ecology as well. Things like amplicon sequencing or DNA metabarcoding. And this is really not that different from sequencing a single gene as we used to do, but we can sequence a single gene on a very large scale. So we can take a sample that's got hundreds of different organisms in it and sequence a gene from all of those organisms at the same time. So this is a method that's been being used for things like eDNA, environmental DNA, where we can take a water sample or a soil sample and sequence a gene like 16S from that sample and get sequences for all of the organisms that are in that sample. So that's one method that's been used, is being used widely, but again, for phylogenetics, it's still just one gene that we would get. We can just do lots of different species at one time. Transcriptomics is sequencing of expressed RNA. So this sequences all the exons, any protein coding gene that is being expressed at a particular time when the sample is taken. And this is in a way we can get sequences for lots of genes simultaneously. It's been used to a limited extent with these corals, limited mainly because it's very difficult to get RNA from them, right? So collecting a coral in the field and keeping it alive long enough to get high quality RNA out of it turns out to be really difficult. So this has not been used that much for corals, but it's being used in many other organisms. And then we have some methods that are based on sequencing just a subset of the genes in the genome, sequencing the DNA. So restriction site associated DNA, or RAD sequencing, is a method that takes a random set of genes from the genome. So we use a restriction enzyme that would cut the DNA at a particular location to fragment the DNA in lots of places, and then we sequence a subset of those fragments. And when you do that, you don't know what you're getting. You're just getting some random subset of the genome, and then you can look for variation in those sequences that you get. This has been widely used now for sort of population genetics and looking at closely related species, but it's got limitations. It doesn't work that well when you have distantly related species. You sequence a random set of genome, you're getting different random things. So you might sequence one species, get a set of randomly selected genes from that species. A different species, you get a different set of genes. So it's hard to make comparisons among distantly related things. The two methods that we've been working with and developed for use with corals in my lab are target enrichment and genome skimming. So I'll talk a little bit more about those and how they work, and so what we've been able to do. So, so target enrichment is also called hybrid capture, and it's a method by which we can sequence a subset of genes in the genome, but we choose what genes those are. And what we're choosing in this case are regions of the genome that are very highly conserved. So these conserved regions can be parts of exons, protein coding genes, or they can be highly conserved regulatory regions of the genome. And we want them to be highly conserved because what we're doing is designing a probe, which is basically the same thing as a primer that you would use for PCR. So it's a complementary piece of DNA or RNA that exactly matches that conserved region. And if it's a very conserved region, we can use the same probe in lots and lots of different species. That conserved piece of DNA shouldn't differ across species that are very evolutionarily distant from one another. So we can design a set of probes that will match and target those conserved regions in the genome. And then what you do is you take the whole genomic DNA, you break it up into small fragments, and then you hybridize these probes to those fragments. So in this example here, maybe we divided the DNA up into 5 fragments and then added the probes, and 3 of those fragments have a highly conserved region that matches one of our probes. So a probe would match this fragment and this one, but not these 2. So once we've matched the probes to those fragments, we use PCR to make lots and lots of copies of them. So this is where the name target enrichment comes from. We target those conserved regions using the probe and then enrich it by making lots of copies. So now our pool of DNA is mostly copies of those conserved regions we want. And when we sequence it, mostly what we'll get are sequences for those conserved regions. Okay? So the question you might wonder is, well, these are really highly conserved regions. They don't differ between species. How is sequencing them going to tell us anything about the relationships of the species we've sequenced? So this technique is based on it's theory, but it's been shown empirically as well, that in one of these fragments, so we say we target this fragment using a probe, and then we sequence the whole fragment, not just the region where the probe binds, but the area on either side of it as well. And those areas are likely to have more variation in them than in the conserved region. So this is a graph. This technique was originally developed for birds. This figure comes from a paper about birds. So on this graph, the x-axis is just the position in this in the fragment, with 0 being like the center of the fragment or the center where this conserved region is and where the probe bound. So, and then this is the amount of variation in that sequence. So here where the probe binds, there's very little variation. But as we move away from the probe in this direction, the amount of variation goes up. And in this direction, also the amount of variation goes up. So by sequencing, starting with the probe and then sequencing out from it, we can sequence a lot of variation that is then useful for understanding the evolutionary relationships of organisms and even population genetics. So we can use the same data at lots of different levels of genomic comparisons. Okay. Another advantage to this technique over some of these others is we can do this with very highly degraded DNA. We don't need high quality DNA. For the other techniques like transcriptomics, for RAD, you need high quality DNA. For this, the first step in the process is to fragment the DNA. And in old specimens, poorly preserved specimens, historical museum specimens, the DNA is often already degraded. And that just saves us a step here. We don't have to fragment it, it's already fragmented. And using this technique, we have now been able to sequence hundreds of, over 1,000 genes from, let's see, I think the oldest specimen that we've done so far is about 190 years. So, it works very well for historical material. So the technique has been developed for birds and then extended to other vertebrates. And in my lab, we extended it to corals. The main work you have to do is to identify those conserved regions. So you have to take existing genomes and align them and find the areas where they're the same and find those conserved regions, and then actually design the probes that are going to match those regions. So we did that in my lab and developed a set of these probes that can be used for octocorals and also for spharectinians and all of the different anthozoans. And the initial probe set we developed targeted about 1,800 of these conserved loci. We've now refined those data sets and are targeting more like 2,000 or 2,500 loci in these different groups. Before I show you the results of that method, let me mention that now we're moving to another method that is actually easier and, most importantly, cheaper than the target enrichment. So the target enrichment is quite expensive because you have to have those probes synthesized to do the enrichment. Genome skimming is a method that's basically just sequencing the whole genome, but you do it at low coverage. So when you do this, you don't get every gene in the genome. Okay? Mostly what you get are things that are already at high copy number in the genome. So things like mitochondrial genes or ribosomal genes that are present in many, many copies. You do genome skimming, you'll get sequences for all of those. Single copy genes, you might or might not. It's highly variable. So this method had been being used recently for as an easy way to get whole mitochondrial genomes, sequence the whole mitochondrial genome, just low coverage sequencing of the entire genome, and then you get all of the mitochondrial genes from that. We weren't sure if this would work for getting anything other than mitochondrial genes and these ribosomal genes, but we decided to try to use it to get the same conserved regions that we had been targeting with the target enrichment. And you do this bioinformatically. So with the target enrichment, you enrich first before you sequence. So you make lots of copies of those target genes and then sequence them. In this technique, you sequence everything, and then afterwards you go in bioinformatically with the sequences of your probes and find any fragments that match those probe sequences. And it turned out it worked really well. We can recover as many loci and sometimes more with the genome skin of these conserved loci with the genome skinning as we were getting with the target enrichment. So this is a tree that we just published last week, in which we combined the two techniques to do this large phylogeny of octocorals. And in this huge, big, unreadable tree, anything here that's black was a sequence we got through the original target enrichment technique. And anything that's blue, the sequences we got with the genome skinning. And again, we get all the same loci and then can combine them all into a single tree. So the genome skimming looks like it's a very nice way to generate data that is then compatible with, it can be combined with lots of other data sets. So you can combine it with target enrichment sequence data, you get the whole mitochondrial genome. We also got most of the ribosomal gene complex, and as well as these conserved elements from doing this. So, and it's about half the price of the target enrichment. Okay, so what have we learned from doing all of this? So this is the first phylogenetic study we did with the target enrichment method, and this is for all Anthozoa. So the tree on the right here is the Hexacoralia class, and then these are the octocorals. And this tree was constructed with about 1,200 loci, kind of these conserved loci. And one thing you can see is there's much better resolution along the backbone of this tree. So now we really are understanding what the patterns of divergence were deep in this tree, and can start to better understand which families are more closely related and so on. We also were able to date this tree. So we can include some fossil calibration points, which are the red dots here, and then estimate the divergence times of these different groups. And what we found is that the anthozoans, all these corals, are much older than the fossil record would suggest. So the divergence of the Anthozoa first diverged about 800 million years ago. So well back in the Precambrian. And even the octocorals arose around 600 million years ago. Scleractinian corals, stony corals, about 400 million years ago. And what that also means is that some of the traits of these organisms are also very old. So if octocorals were around 600 million years ago, all octocorals form colonies. So the trait of being able to make a colony must be very old. We just recently published a paper looking at the evolution of bioluminescence in this group. Many of the deep sea octocorals can produce light, bioluminescence, and that dates back to about 500 million years ago, much older than people had thought. So with this well-resolved tree now, we've solved a number of phylogenetic questions about relationships to the group, but also beginning to understand when the traits in this group evolved. Just to show you the first trait that we addressed was the issue of a skeleton. And again, the composition of skeletons is something that's very important right now ecologically, because with climate change and changing environment, and in particular ocean acidification, we know that ocean acidification is going to affect the ability of organisms to produce calcium carbonate, and the form of calcium carbonate that they have, or the amount of calcium carbonate that they have, is going to determine, how they're impacted by this ocean acidification. So we wanted to look at the evolution of different types of skeletons in these anthozoa. How many times have different skeletal types evolved or been lost? When did they occur? This is the same tree that I showed in the last slide, but turned into a circle so that it fits on the slide better. So you would read it, if we straightened it out, this would be the right side of the tree, no, sorry, the left side of the tree, around to the right side of the tree. So everything at the top here are octocorals, and everything at the bottom are the hexacorals, and the different colors are different types of skeletons that have evolved. So the gray you see down here are all sea anemones that don't have skeletons. So they had no skeleton. And then we see in the hexacorals, the evolution here, the pink is the evolution of a proteinaceous skeleton in the black corals, which looks like it just happened once, and all black corals have a proteinaceous skeleton. They've never lost that trait. These are the scleractinians in blue. Again, scleractinians evolved a big skeleton of aragonite once. None of them have lost it, so they all have that. This is the octocorals, right? Rainbow. The different types of skeletons in the octocorals have been lost and gained lots of different times. So we can see things like the yellow color here. It's a lot of yellow in this group here, but yellow has evolved multiple times within this group, and we see some yellow over here as well. So the same type of skeleton has evolved in those two different major groups of octocorals. The same is true for things like, I don't know if you look at the green, which is one of those types of axes made out of these sclerites that have been cemented together. You see it occurring here and here and here and here. So octocorals have reinvented the same skeleton type many different times in different groups. And they've also lost a skeleton. So orange are soft corals, things that don't have a skeleton. And we can see out here in this group, which is mostly gorgonians, there are some orange branches showing up. These are soft corals that apparently evolved from gorgonians through the loss of the skeletal axis. So compared to the scleractinian corals, which invented a skeleton once and stuck with it, the octocorals have evolved different skeletons many different times and have lost them as well as gained them. And we've done the same thing with other traits like photosymbiosis, so the ability to have zooxanthellae and gain from the photosynthesis. Same thing, it's evolved many times in all of these different groups, been evolved and lost. It's a trait that again has been reinvented many times. So the other thing that we're now in a position finally to do was some major taxonomic revision. And I did this about 2 years ago for the octocorals, changed the names of everything, so everybody hates me now. But we finally thought maybe the taxonomy should reflect what we understand about the evolutionary relationship. So the three existing orders, Alcyanacea, Penatulacea, Helioparacea, gone. Helioparacea got demoted to be a family. The sea pens got demoted to be a superfamily, and we now have two new orders that reflect those two big clades we were seeing. So one is the Scleralcyanacea. This is this group here, which they said is mostly deep water things, and Scleralcyanacea means hard alcyanaceans, hard octocorals. So these things, what they have in common is most of them have a pretty solid calcareous axis structure. So this includes the blue coral is in this group with its aragonite skeleton, the sea pens, which do have an interior axis with a solid calcium carbonate rod, various families of deep sea gorgonians that have, again, solid calcareous skeletons. So that's the majority of this group. There's exceptions. The other group we call malacalcinacea, which means soft alcinaceans. And this includes, again, mostly shallow water taxa, most of the gorgonians that have a protein axis rather than a calcareous axis. Most of the big fleshy soft corals and the stoloniferans are in that group. So that's our new classification. And we also erected a lot of new families. So those cases where one family was divided up into like 6 or 8 different clades throughout the tree, many of those are now new families. So shifting to looking at, again, how this new phylogeny and understanding of evolutionary relationships impacts our understanding of how traits evolve. I want to mention 2 different families that were extremely surprising to us. So this is an example of a very complex morphology that appears to have evolved independently in some very distantly related lineages, and this was a total surprise. So this is a type of skeleton that we find in some gorgonians, where we have segments of solid calcium carbonate that are separated by joints made of protein. And we call these bamboo corals. Okay? And all of the bamboo corals have been put in a single family, and it was assumed this was a trait that had evolved only once. It's really unique and sort of specialized. But it turns out the bamboo corals occur in 3 or 4 different families that aren't a clade. So this is a typical, these are common in the very deep sea. And here's where the family of the deep sea bamboo corals is. There's another deep sea one that's up here that again doesn't share a common ancestor with this one. And then down here in the other order, there's this one, which is actually a shallow water bamboo coral. And its closest relative is something that looks totally different. It has a normal proteinaceous axis. So this highly specialized skeleton type has clearly evolved multiple times in these different groups. Okay. Another family that was a big sort of surprise to us, this group called the Coralliidae, which we see up here. It's this yellow group, a nice group of things that are all pretty closely related, fall into Coralliidae. And the species that are now in Coralliidae had previously been put in 3 different families, because this is what they look like. So this is this is corallium, which is the precious coral they use for making jewelry. And these are large fan-like colonies that have a very hard skeleton that's made of sclerites that are fused tightly together with calcium carbonate. And if you have any coral jewelry, you know it's a very hard substance. So they were in one family. This was in a different family, Paragorgia, family Paragorgiidae. This is a deep sea group that are often called bubblegum corals because of their color. And again, the form of the colony looks kind of similar to this, but the skeleton is very different. They don't have a hard skeletal axis. It's got those sclerites in it, but the sclerites are embedded in sort of protein and things. So it's a very soft skeleton, very different from this one. And then there's these things, which were in a 3rd family. These are soft corals. They don't have an axis. These little mushroom-shaped things. And it turns out all of these are really closely related, which we never anticipated based on what they look like. So we now had to ask, particularly in doing this taxonomic revision, well, what makes a coralliid? If their form is so variable, is there any morphological character that these things share that would unite them in a family? So we've had to start looking at different characters, and what we found is where the macromorphology doesn't inform us about the evolutionary relationships, the micromorphology does. So if we look at the sclerites, these microscopic calcium carbonate crystals in the tissue, here's a sclerite from the corallium, Paragorgia, and these little mushroom things. they all look very similar, and they're quite different from the sclerites that you find in other families. So this is a shared characteristic of this group. It's also the case that everything in this group has what are called dimorphic polyps, two different types of polyps, one for feeding and one for pumping water through the colony. There's some other groups that have that, but all of the species in this group have that characteristic. So through looking at those sorts of characters, those are things that unite things as belonging to this family Coralliidae, despite the fact that their colony morphology and skeletal form is really, really different. So we've had to reconsider what morphological characters are phylogenetically informative, and it's also suggested to us what morphological characters are very evolutionarily labile. That is, they change evolutionarily quite quickly. So I'm gonna shift just for the last few minutes here to species boundaries questions, which is what I spend most of my time worrying about. So it said these sclerites are found in most species in the soft tissues, whether the species has an axis or not. And these are the primary morphological characters that have been used for a taxonomy at the species level. So if we want to know, and so families have specific sclerite shapes, genera have specific sclerite shapes, and at the level of families, also, or sorry, at the level of species, also the shape, the size, the distribution in the tissues of these sclerites is what we use for determining if things are different species or not. And it's very hard because these are highly complex shapes. They're highly variable. No two sclerites are exactly the same. And they're hard to quantify and hard to image. So in the recent times, so since about the 1990s, with scanning electron microscopy, if we describe a new species, we will take scanning electron micrographs of the sclerites and get these very nice high resolution images that we can then use for identifying that species. But that's only been for the last 30 years. And before that time, when people describe species, if they even illustrated the sclerites, they hand drew them. So what we're stuck with a lot of times now is trying to identify a species. We've got something that looks like this under our microscope, and we have to decide, is that the same as this? Which makes it almost impossible to put a species name on almost any octocoral out there. And ecologists get super frustrated. Ecologists want names, right? Tell me what it is. I want to know what I'm working with. And say, I don't know. You know, it could be one of 20 different things, but I can't tell if what I'm looking at right now under my microscope is the same as this or something else that's been drawn kind of similar to that, and so on. So we thought that molecular techniques was going to be our savior, and being able to do things like DNA barcoding, where we can take a sequence of a single gene, and if we know what species that gene came from, then we can use that as an identifier, right? So just sequence it, much easier than this. Sequence it, okay, look at the sequence, match it to some database, got the ID, right? So that's the whole idea behind DNA barcoding, and it works really well for birds and butterflies and fish and things like that. We know what sequences, identify different species. But corals have been a real nightmare because of what's called the anthozoan problem. So usually for DNA barcodes, we use mitochondrial genes because in most animals, they evolve 5 to 10 times faster than the nuclear genome. Which means that even closely related species have different sequences at those mitochondrial genes. Anthozoan corals are the exact opposite. The mitochondrial genome evolves 10 to 100 times slower than the nuclear genome. So most of the mitochondrial genes in corals are not variable enough to distinguish closely related species or sometimes even genera. So DNA barcoding has not worked for us very well. And I've spent about the last 15 years searching for the better barcode, the perfect barcode, and haven't found anything. So we've estimated that we can identify maybe like 80% of things with barcodes, but that's 20% of things where it's like, I don't know, there's about 3 or 4 different morphologically different things that have this exact same sequence. So it doesn't tell us what they are. So we're hoping that these newer techniques, these phylogenomic techniques, are also going to help us with identifying species and figuring out what's really what out there. So we've done some testing of these different methods to discriminate species. And this is one example with a group. I work a lot on a group of a genus of soft coral. They used to be called Sinularia. I changed the name with a revision. It's now sclerophytum. And it's about 180 species. They're very common on Indo-Pacific coral reefs. You often have as many as 30 species on the same reef. You can't tell them apart morphologically, at least not reliably. And this is using some of the barcodes. So this is 2 mitochondrial gene and a ribosomal gene that we developed as a barcode, and using that to try to distinguish about 10 or 12 species in this group. And it works fine for some. So you can see this species and this species and this species are quite nicely separated with these barcodes, but others aren't. And in particular, in this group here, there's this species Sinularia acuta, which is showing up here mixed with 2 other species, and here mixed with another species, and here mixed with another species, and so on. So again, these barcodes work fine for some species, but not for others, because we have things we think are different species that have the same barcode, or we have one species that has several different barcodes. So this is now taking the same individuals and doing the target capture to get these conserved elements, so getting over 1,100 loci rather than just 2, and now looking at them. And now we separate the species out quite nicely. So these methods are working for at least helping us decide what the different species are. It's still moving forward. It's going to be hard if you go out and collect something on the reef and want to know now, okay, well, which one of these it is. Doing this genomic technique to try to do that is still going to be hard and expensive. We've also looked at whether these same conserved elements that we get with the target enrichment or the genome skimming work at the population level, because we haven't been able to do population genetics, particularly with anthozoans, because we don't have genes that are variable enough to distinguish populations. So the study of population genetics of corals is 20 years behind the study of population genetics in most other animals. So in this study, we looked at 4 different populations of a soft coral that occurs in Europe, occurs in both the Mediterranean and the Atlantic. So we had 2 populations from the Mediterranean and then populations from Portugal and France in the Atlantic. Used the exact same genes that we used for that phylogeny of the whole group. In fact, I think some of the individuals in this study were in that big phylogeny, and used some techniques being used for doing population differentiation that are based on looking at single nucleotide polymorphisms in those genes. And we find nice differentiation of populations. So this population in Portugal is very nicely distinguished from the population in France and from the populations in the Mediterranean. It looks like there's some gene flow and admixture between the populations in the Mediterranean, which we would expect because they're close together. But we're getting nice genetic signal at the level of populations, which is something that we haven't been able to do very much in corals for a long time. So, and what my lab is focusing on right now, we are nearing the end of about a 4-year project focused on this group of soft corals that are common in the Indo-Pacific. So this is a group of about 30 or so genera, about 600 names. How many species that actually is, we really don't know. And that's the goal of this project, is to use these phylogenomic markers, these methods where we can get 1,000 genes to try to determine how many species of these shallow water zooxanthellate octocorals, which are very common and often ecologically dominant on Indo-Pacific reefs, how many species are there out there? What are their distributions? How many of them are endemic to particular regions versus have widespread distributions, and ultimately try to put names on them. And we are in the middle of that project right now. But we're learning a lot about the biodiversity and particularly about the distributions. A lot of these things that were assumed to have kind of global distributions or all across the Indo-Pacific don't. They have much narrower geographic ranges than we had thought, and there's a lot more endemic species out there than we had thought. So that's what we're working on now. So hopefully with these newer techniques, we can move ahead with addressing a lot of interesting biological and ecological questions with this group that we have been unable to address just because we don't understand who's who and how they're related to one another. So I think we're at a point we can start looking in the genome, looking at individual genes for the basis for this lability in morphological evolution. How are octocorals able to change their skeletons so easily, evolve new skeletons, lose skeletons, and so on? Does this give them an adaptive advantage as the environment changes? What's the distribution of genetic variation across space? Again, you said it looks like many of these things are not nearly as geographically widespread as we thought, and again, the implications for conservation are high. And again, we're beginning to understand a lot more about the history and the mechanisms by which this process of diversification in anthozoans has occurred. So I'm going to stop there, and I just want to thank 3 people who I've worked with very closely over the years and have contributed a lot to this. So Andrea Petrini was my postdoc, she really developed the target enrichment probe set in my lab. She's now curator at the Smithsonian Institution. William Van Offwagen is a taxonomist who was a close colleague of mine for many years. So they passed away a couple of years ago. And then Yehuda Yahoo at Tel Aviv University is a very close collaborator and my partner in this project to try to sort out all these Indo-Pacific corals. So they have all contributed hugely to this work over time. And I've had an awful lot of undergraduate students in my lab in California who have done a lot of sequencing over the years and contributed in that way. So thank you, and I'm happy to answer any questions if you have questions about any of this.
Thank you very much. Wonderful. Very complex. Complex study. Wow. It's great. I have a question about when you was talking about the sclerites, the soft corals, and try to understand the environment influence that is not well studied yet. What happened? It's very interesting. I am very interested about this. And what about the Promonestia? The Brazilians, what do you think? Do you have some suggest something about it?
Suggest about it's specifically what about sclerites? Or,
yeah, because they are invasive.
Yeah.
And
so can we destroy them by?
Yeah.
Is that something that?
They are in a held carbon that we have a dwelling system there. And now they are in the big island.
Yeah.
So different place. We suggest someone. They took some other.
Well, they just showed up.
But they are with different colors.
Yeah. So the color. So the sclerites. So sclerites are very species specific. The chromonepthia you have here have very complex sclerites and a lot of different types of sclerites. And they're often used for defense. So they're pretty prickly, right? The chromonepthia, some of the other ones that they're related to, you touch them and they're spiky.
Yes.
So they use those for defense. What we know about sclerites, so the color is in the sclerites. Okay, so when you see different colors in chromonepthia, anyway, it's the sclerites are different colors, and the colors are iron pigments that are in the calcium carbonate of the sclerites. And they can be highly color polymorphic. Many octocorals species can come in lots of different colors. It's just a polymorphism within species. We know very little about how the environment influences the form of the sclerites, and that's something else that complicates our ability to distinguish species. We see variation in the sclerites between two colonies. Is that because they're different species, different genotypes, or we're affected by the environment differently? We don't know. So ocean acidification is a problem that is affecting calcium carbonate. The sclerites are made out of a form of calcium carbonate, it's high magnesium calcite, that is the most resistant to acidification. So they are going to be affected less than the stony corals that have aragonite skeletons. And there's experiments that have been done that suggest that you put just loose sclerite, take the sclerites out of a octocoral, so you just have the calcium carbonate, put it in acidic conditions, and they'll dissolve. But you can grow the octocoral in those same acidic conditions, and the sclerites are fine. So we think that the tissue buffers the internal pH of the sclerites. So octocorals may be affected less by ocean acidification. Stony corals, the skeleton is outside the animal, right? It's external. Octocorals, it's inside, and the tissue can protect it from the acidity. So as ocean acidification happens, I don't think it's going to kill the chromonepthia for you.
No. It's good to with berry, so we have lots of options.
Yeah.
Thank you very much.
And I have a curiosity because what you showed us, you have been revolutionizing
That's right.
coral taxonomy.
Mm-hmm.
And have you faced or have you been facing any rejection, resistance by other taxonomies?
Oh yeah. Well, it's very funny. So I published this paper where we got 2 new orders and 18 new families and really radically restructured other families. So Coralliidae, which was just the precious corals, now has these other things in it. I changed Sinularia to make it sclerophytum reasons. And when I published the paper, almost immediately I got all of these sort of congratulatory emails from ecologists saying, "Oh, thank you for finally doing this." It took a while longer before I got emails from taxonomists. And a couple of the big important taxonomists, after about a week, emailed me and said, "Okay, I've digested what you've done, I think this is really good. This is what was needed." And there's 1 or 2 others that I've never heard anything from.
Oh my God.
Yeah. And on the basis of a paper that was reviewed recently, I know one of them is not very happy about the changes. So that's always the case. One of a change that happened as a result of revisions to the taxonomy of the stony corals based on genetics. So in the deep sea, there's a stony coral called Lophelia, which forms big reefs, huge reefs. So there's a huge literature on Lophelia reefs. And a study was published a few years ago using genetics that they discovered that Lophelia, which is a colonial scleractinian, forms huge reefs, was genetically almost identical to another species called Desmophyllum, which is a solitary cup coral. They're so close, their mitochondrial genomes are almost identical. And Lophelia got renamed Desmophyllum. The deep sea biologists still haven't accepted that one. Yeah. So anytime you like do these things or change names, there's a lot of resistance from certain communities. Yes.
I do have a question. It might be rubbish. I don't know anything about corals. I work with dolphins and masks. But do you think like they have all this convergent evolution and also very, when you say that families were put out together and put back together depending on the traits and everything? Do you think it comes to me like plants comes to mind because they are sessile, and they suffer more pressure from the environment than animals that can move? So do you think that played a lot in, because they're a very old lineage as well, like played a huge role in this craziness of traits that you see?
Yep, absolutely. Yep, there's lots of convergent evolution. And the similarity of plants is something I've been emphasizing in the course this week with students, right, Carol? That in their ecology, probably in their evolutionary biology, corals are much more like plants than they are like animals. Yep. And they're, a very, very old lineage. So even, so we see lots of convergent evolution in form, skeletal type, even in the scleractinians where they evolved a calcium carbonate skeleton once, there's still a lot of convergence in that group in form. So about half of the scleractinians are colonial, form big reefs. The other half are solitary, and they're just what they call cup corals, so single polyps. And that's a pretty big difference between a cup coral and a colony. But we're learning more and more. I'm learning from other people's work more and more that, again, the cup corals have evolved lots of times within the scleractinians. So in the scleractinians, there's a family called the Caryophyllidea, which is kind of similar to our Alcyonidae, where Caryophyllidea are little cup corals that thought they were all one family, and no, they're all throughout the tree. So again, that very similar morphology has evolved by convergence lots of times.
Another curiosity is that do they have like sexual chromosomes, or they are like plants as well? In that sense, that they can hybridize. New species can come from hybrids.
That's a very interesting question. So it has been thought that there's a lot of hybridization in corals. We've shown through genetic methods that yes, there are signatures of hybridization in the past in species. Some interesting work that's just been published recently, I'd say about 20 years ago, all corals are just wildly hybridizing, and that's why we can't tell them apart, because they're like all this hybrid lineages. So with the more recent genetics and ability to look in more detail, it's looking like hybridization is maybe less common than had been thought at that time, but we still have some very good examples of hybridization occurring. So yes, they're like plants in that as well.
Sorry for my English. The conservative parts of the DNA used in the technique enrichment, I forgot the name. These parts, the genes, what they express. Do you know what they express?
We know some of them, not all of them. So many of them are various different proteins. So they're highly conserved parts of the exons of proteins. And we've been able to identify quite a number of them. I can't tell you off the top of my head what gene families they belong to, but they are protein coding genes. Others look like they're probably not protein coding genes, but they're regions of the genome involved in gene regulation that are just highly conserved. And that's originally, there's a term called UCE, ultra conserved elements. And those were originally thought to be mostly or entirely regulatory regions. But we've been discovering that some of these genes that we thought were regulatory regions actually are exons. They're from the protein coding. So it's a mix, the things that we're looking at. Some are protein coding, some are regulatory. What they have in common is they're highly conserved.
I have one question about the if you are trying to identify a species by barcode, for example, but each species was never listed before, so there is no data about it on the project.
Yeah.
Can you locate the sequence in the exon tree, or it's a kind of possible?
So say let me understand, if we have something we don't know, we haven't sequenced it before,
Yeah.
can we sequence it and then figure out where it is in the tree?
Yeah.
Oh yeah, I do that all the time. I mean, yeah, people send me stuff, they don't know what it is, we don't know what genus it is, whatever. Just sequence it, then we'll figure out where it goes. Yeah. So, yeah.
Have you ever worked with freshwater coral?
There are no freshwater corals.
Oh,
Yeah.
nice.
So the anthozoans are entirely marine. No, wait. There's some anemones that occur in brackish water.
Yeah, because
Yeah.
in the estuary, sometimes I've been doing field work, and sometimes you have people working with fish, and they put nets to capture stuff. And sometimes they pull up interesting stuff. I think they're corals, but again, I know nothing about those things. Anything that it's like seashell and
Yeah, they might be in brackish water.
yeah,
But
yeah,
not-
the estuary
yeah,
is on,
yeah.
the Amazon estuary.
Yeah. There are other groups of cnidarians, like the hydrozoans. There are a lot of freshwater hydrozoans and freshwater jellyfish. And I don't know, the hydrozoans also form corals. There's what are called hydrocorals. But I'm not sure any of those are in freshwater, but maybe they.
Yeah, the things that I've seen there are in
Brackish.
brackish,
Yeah.
yeah. But I don't have any pictures of
Yeah.
that, but I do have pictures.
Hmm. Yeah.
Yeah, it's not in the bloom, because the bloom, it's when you go out of the Amazon and just like that. So it's not, it's more inwards. It's, have you ever heard of Barcarena? Yeah, where they do all the Amazon. Yeah, so there we found this like coral-like things. They look like it, but I didn't. But I do, they found reefs in the outer waters of the estuary as well. But again, like it's super, like during the rainy season, it's very fresh water. Like if you go far into the sea, it's very murky because of the water of the Amazon comes with a lot of water. That's very.
Okay. Thank you very
Well,
much.
thank you.
Thanks a lot. It was very
Thank you. This was